So in chapter 13, we're going to be talking about blood and alterations in oxygen transport. So just starting out, we're going to talk about the normal composition of blood. Um, Total blood volume in men is about 75.5 mils per kilogram, and in women, 66.5 mils per kilogram on average. And so therefore encompasses about 7 to 8% of your total body weight. The average blood volume is about 5 to 6 liters, and blood cells within a particular volume of blood make up about 45% of that volume. Otherwise, the remaining volume of blood is plasma, which is mostly water, plasma proteins, and electrolytes. So we're going to talk about how blood first forms, and it's a two-stage process that involves cell division or mitosis, as well as maturation or differentiation. Now, the hormone that stimulates mitosis or proliferation of red blood cells is a hormone called erythropoietin, or EPO. And this particular erythropoietin hormone comes from the kidney, and it stimulates erythrocyte production within red bone marrow. So what happens is the kidneys will sense when blood oxygen levels are low, they'll release erythropoietin in order to stimulate red bone marrow to make more red blood cells. And then when you get more red blood cells, blood oxygen levels may rise and bring you back to homeostasis. Now you might wonder, well, how does blood carry oxygen? Well, remember, about 45% of the volume of blood is red blood cells, and within each red blood cell, you have about 250 million molecules of hemoglobin. And so what this slide is showing is one hemoglobin molecule. You can see that it's actually made of 4 separate proteins that are all stuck together to form this hemoglobin structure. And then within each of these globin chains, so there's 1, 2, 3, 4 globin chains. Then you have a hemoglobin here, a heme group in the center of each of these globin chains. Now the heme group has a central iron atom, and it's the iron here in heme that's actually binding to gases like oxygen, carbon dioxide, and carbon monoxide. So this heme is essential to play a role in the transportation of gases. Now, every single red blood cell has an average lifespan of about 120 days, so about 4 months or so. They don't last forever. And one of the reasons why red blood cells have such short lifespans is they really lack most organelles. Red blood cells don't have a nucleus, and they don't have other organelles to help maintain the cell's integrity over time. So when they start to degrade, they have a shorter lifespan, so about 120 days or so on average. It turns out that old or defective and damaged or dead red blood cells or erythrocytes are removed by your spleen and liver. And there's actually a filtering action here where mononuclear phagocytes, so like Kupffer cells, that line the inside of blood vessels within your spleen and liver can sense whether a red blood cell is damaged or not. And if it encounters a damaged red blood cell, it actually will phagocytose and remove that from the bloodstream. So the feedback loop here for new red blood cell production is a bit of communication between red bone marrow and your kidneys. So what's interesting then is that the amount of red blood cells in your bloodstream is going to correlate with how well you're able to transport gases like oxygen. Now it turns out that the kidneys have oxygen sensors, and so when O2 levels get too low in the bloodstream, so if you have hypoxemia, it turns out the kidneys can release a hormone called erythropoietin. And this erythropoietin hormone will travel through the bloodstream, make it back to red bone marrow, and then stimulate erythrocyte production. And when you produce more erythrocytes, you'll increase your red cell mass and potentially get your blood oxygen levels back to a homeostatic level. Once blood O2 levels are at homeostatic set point, the kidneys will not release as much erythropoietin, and therefore you don't make too many red blood cells. So this is showing that feedback loop here. But what's interesting about this form of communication is that if the kidneys are failing, you may see a situation where they can't release erythropoietin, which means you have difficulty stimulating more erythrocyte production, and that may lead to anemia. So there's different forms of anemia that can result from things like kidney failure. And so what this slide is showing is just normal gas transport and acid-base balance in your body. We know that our lungs here play a role in the transportation of gases into and out of your body. So if you look here in this diagram, we'll first start at this side. And what this is showing is the right side of the heart. So you're going to have like the right atrium and right ventricle, and they're going to pump blood towards the lungs. Now, for the blood that's going towards the lungs, we call this deoxygenated blood. It's the partial pressure of oxygen in blood going to the lungs is only 40 millimeters of mercury, whereas a fully oxygenated blood could be somewhere around 100 millimeters of mercury. So it's sort of a misnomer to call it deoxygenated blood because you're never actually going to fully run out of oxygen, but it has a lot less oxygen than you could have. So for that blood going to the lungs, we call this deoxygenated. Well, what this means if it's deoxygenated is that there are less sites on hemoglobin that are bound to oxygen. So when that blood reaches the lungs and it starts to go through the capillary beds surrounding the alveoli or microscopic air sacs in your lungs, that has the potential to pick up atmospheric oxygen from those spaces. So therefore, when the blood leaves your lungs then through pulmonary veins, it has absorbed a lot of oxygen already, and it leaves the lungs in its oxygenated form. So you can see here that we've increased our oxygenation by twofold or more. And this oxygenated venous blood goes back to the left side of the heart, where the left atrium and left ventricle then will pump that blood out to the rest of your body. Now, what happens next, you guys, is that we get internal respiration. And internal respiration is where you get the exchange of gases between blood and your body's tissues. Because your body's tissues use oxygen to make ATP, what will happen is they're going to start to absorb oxygen out of the bloodstream, and therefore when blood goes through capillaries and you get that sort of internal respiration or gas exchange, that oxygen starts to be depleted, and that's when it turns back into what we call deoxygenated blood, and then it travels back to the heart, and the whole process repeats. Now, what's interesting, though, is that hemoglobin has some interesting properties. What this is showing is the oxygen hemoglobin dissociation curve. And what this shows is a relationship between oxygen saturation on hemoglobin and the partial pressure or concentration of O2 in blood. So if we look here, then we find that there's a positive correlation between oxygen saturation on hemoglobin and how much O2 you can find in the bloodstream. And that kind of makes sense. If your hemoglobin's carrying more oxygen, then there's the potential to release more oxygen in the bloodstream. And that's what we see is that sort of trend here. But what you'll notice is that it's not a linear relationship. We actually get these different forms of like an S curve here. And these S curves can actually change depending on different variables. So if you look here in purple, or kind of pinkish, in curve A, what we find then is that under this particular condition in curve A, we find that hemoglobin actually holds on to oxygen more strongly, because even when it's 70% saturated, you have approximately 25 or so millimeters of mercury of oxygen available in blood. Now let's compare that to another curve. So let's look at B. Let's see when curve B hemoglobin is 70% saturated. This one is actually about 35% or 35 millimeters of mercury or so of oxygen available in blood. So what this shows then is that between curves A, B, and C, it's a relationship of how hemoglobin has varying affinities for oxygen. So with curve A, this is the conditions that promote hemoglobin to hold on to oxygen more strongly and be less likely to release oxygen under changing levels of oxygen saturation. B is going to be a condition where hemoglobin releases oxygen more readily, and therefore at different degrees of oxygen saturation, you can find higher levels of O2 in the blood, and C even more so. And so we can talk about left shifts and right shifts. If a left shift occurs, this means hemoglobin will hold on to oxygen more strongly and be less likely to release oxygen into the bloodstream. If a right shift occurs, this means hemoglobin will be more likely to hold on to oxygen, less likely to hold on to oxygen and release that oxygen into the bloodstream more readily. So what are the factors that induce a left shift or a right shift? Well, if we look at left shift factors over here, some factors that can lead to a shift to the left could be like decreasing hydrogen ion concentration, so making your blood pH less acidic, which would be an increase in pH. So if your blood pH becomes less acidic or more basic, like if you go from 7.35 to 7.5, that could induce a left shift. Now this also correlates with CO2 levels in the blood, because if y'all remember going back to A&P, when carbon dioxide dissolves in blood, it forms an acid called carbonic acid. So if CO2 levels are low in blood, then your blood's less likely to be acidic. Another condition that could affect a left shift is a decrease in temperature in that blood going through a tissue. So if the blood becomes cooler, then hemoglobin is less likely to release oxygen in the bloodstream, so you get a left shift. Now, some other factors too would be hormones like 2,3-DPG, which is released under certain conditions. There are other congenital disorders as well that can lead to left shifts, like hemoglobin Rainier, hemoglobin Hiroshima, and hemoglobin San Francisco. These are all congenital and heritable disorders of hemoglobin, where hemoglobin is less likely to sort of release its oxygen into the bloodstream. Now, right shift factors are basically just the opposite. So something that would make hemoglobin more likely to release oxygen in the blood would be an increase in hydrogen ion concentration, which correlates with a decrease in pH. So that if you get more hydrogen ions in blood, this makes blood more acidic and therefore has a lower pH. This also correlates with CO2 levels, because remember, CO2, when it dissolves in blood, can form an acid or carbonic acid. So with rising CO2 levels, this makes blood more acidic here. So CO2, hydrogen ion, and a decrease in pH, these all interrelate. Also an increase in temperature. So if blood goes through a warmer tissue, and therefore the blood warms up, an increase in the temperature of that blood will actually cause a right shift. And you might wonder, why is this? Why would you want hemoglobin to have left shifts or right shifts? Why, under what circumstances would you want hemoglobin to left shift and be less likely to release oxygen into blood? Well, let's think about a condition where, what if you have tissues in your body that are more metabolically active than others? Let's say if you are exercising and the blood that's going through some of your digestive organs is, potentially maybe a little cooler, because at this period of time your digestive organs are underactive, so maybe your internal body temperature is a little cooler. This may induce a left shift, because you don't want to release oxygen into a tissue that doesn't need it. So instead, that hemoglobin can hold on to its oxygen until it encounters a warmer tissue like skeletal muscle that's currently active. And once that hemoglobin encounters the warmer tissue, it can undergo a right shift, which could be due to an increase in temperature, and therefore be more likely to release that oxygen into a tissue that needs it more. So this whole idea between left shifts and right shift relate to the fact that not all tissues in your body need equal amounts of oxygen, and these are the factors that can influence whether or not hemoglobin is more or less likely to release oxygen into a tissue. Now, for carbon dioxide transport, earlier I mentioned how CO2 can dissolve in plasma, but can also be converted to carbonic acid here. So what we find then is that for a certain proportion of CO2 that gets absorbed by your blood, some of it is actually converted to carbonic acid by an enzyme called carbonic anhydrase. Then this carbonic acid will dissociate into bicarbonate ion and hydrogen ion. So what's interesting is the way you transport CO2 in blood is in the form of bicarbonate ion, which came from carbonic acid, and it turns out this is actually what baking soda is made of, surprisingly. So 75% of the CO2 you find dissolved in blood is actually transported as a bicarbonate ion, which is one of the components of baking soda, which is interesting. That's not to say you should inject baking soda in your bloodstream, but it's just interesting how this molecule is transported in that form. Now, some CO2 actually binds directly to hemoglobin, and it forms what we call carbaminohemoglobin. And this is where CO2 can just bind directly to hemoglobin, and about 20% of your CO2 is transported in that form. Otherwise, a smaller percentage of it just dissolves directly into blood plasma as a gas. Now, what we'll move on to next talk about are the conditions of altered oxygen transport, which is really what this chapter is all about. So the first portion was just a review of A&P. And we're going to first talk about this concept of anemia. So anemia can come about from a deficit of red blood cells, which causes a low oxygen carrying capacity. So if in your body you can't carry a significant amount of oxygen, this could lead to tissue hypoxia, or low O2 levels in tissues of your body. Now, the opposite of anemia is called polycythemia, and this is due to an excess of red blood cells. And so in previous chapters, we talked about how there was a condition called polycythemia vera, which was a cancer of the stem cells that create red blood cells. But polycythemia means an excess of red cells, and you might wonder, well, why is this a bad thing? Well, too many red blood cells in your bloodstream can make your blood more viscous or more resistant to flow, and also increase blood volume to the point where you could have high blood pressure. Now, there are two main types of anemia. We have relative anemia and absolute anemia. Now, relative anemia is about where the total number of red blood cells are going to be sort of diluted out with changes in plasma volume. So with relative anemia, you haven't changed the total number of red blood cells. However, what can happen though is that if you get increases in plasma volume, then you're sort of making your red blood cells more dilute. So I'll give you guys an example. Let's say if you drank a tremendous amount of water and that water was absorbed into your blood, well then you're essentially increasing the percentage of blood plasma. So whereas normally you should have about 55% of your blood plasma blood volume being that to plasma, if you drink a significant amount of water, that percentage can increase more so, which means you relatively have a lower percentage of red blood cells in any volume of blood, and that can lead to relative anemia by sort of diluting out the existing red blood cells. Now, this differs from absolute anemia because absolute anemia is actually due to a decrease in the number of red blood cells, and this can come about from a variety of mechanisms like decreased production of red blood cells or increased destruction. And we'll next talk about the disorders that lead to either decreased production and increased destruction that lead to absolute anemia. So for the general effects of anemia, what we find then is that just first of all, you're going to see a decrease or deficit in oxygen carrying capacity, which will lead to tissue hypoxia. Now, there are some compensatory mechanisms to restore tissue oxygenation. You'll see things like an increase in heart rate, an increase in cardiac output, circulatory rate and flow to vital organs in order to make sure that you can receive enough oxygenation up to those organs. You'll see that red blood cells will release more 2,3-DPG, and this is going to help decrease oxygen affinity to hemoglobin and therefore help release more oxygen into the bloodstream. So this would induce a right shift. And you're going to see also an increase in erythropoietin activity because when the kidneys sense low O2 levels in blood, they'll release erythropoietin and try to reverse the effects of anemia. Now, mild anemia usually will result in no clinical symptoms. You can find this in individuals like the elderly or individuals with cardiovascular pulmonary disease may also have symptoms of mild anemia. Now, mild to moderate anemia can lead to things like fatigue, weakness, loss of stamina, or tachycardia. And so this would be due to basically just low O2 levels, so deficit of metabolism throughout your body. Now, moderate to severe anemia can lead to things like orthostatic hypotension, which is actually low blood pressure upon standing, vasoconstriction, pallor or sort of whiteness of the face, tachycardia or fast heart rate, murmurs, angina pectoris, which is intermittent heart pain, or even complete heart failure. This can also lead to things like night cramps, headache, lightheadedness, faintness, tinnitus or ringing in the ears, and these can all be due to moderate and severe anemia, which in the case of heart failure, which means that it could become fatal if it's severe enough. So what are the things that cause anemia? Well, the first type of anemia we'll talk about is aplastic anemia. Aplastic anemia is a stem cell disorder that's characterized by a reduction of hematopoietic tissue. And so we find that it is actually a replacement of red bone marrow with yellow bone marrow or fatty marrow. And so what we see then is that not just a deficit in red cell production, but also low white blood cells and platelets too. And this can be caused by toxins, radiation, or other immunological injury that really causes damage to your red bone marrow. It's easily diagnosed with bone marrow biopsy, and it's often a disease of the young or old. Now, anemia of chronic renal failure is where you can get basically anemia due to the fact that if your kidneys can't release erythropoietin, then they can't stimulate a compensatory increase in red blood cell mass. So anemia can result from renal failure by the deficit of the erythropoietin cascade. Now, there's also anemia that relates to vitamin B12 deficiency. Vitamin B12 is necessary for DNA synthesis. So if you have a diet low in vitamin B12, which is common in like vegetarians and vegans, this can lead to anemia in that regard because you need to make DNA in order for cells to divide. If your cells can't divide, then you can't make new red blood cells. And there's a form of anemia called pernicious anemia, which is actually due to a lack of intrinsic factor in the stomach, which causes vitamin B12 deficiency in your body. And this is not to say if you don't have sufficient vitamin B12 intake, but rather if your stomach can't make intrinsic factor, even if you're ingesting enough vitamin B12, you won't absorb that vitamin B12 because you need intrinsic factor to absorb it. Iron deficiency anemia results from nutritional deficiency in iron. It's the most common. We know that iron is necessary for hemoglobin synthesis, and so what we see then here is hypochromic and microcytic red blood cells because they're going to have a lack of hemoglobin, and therefore the cells won't look as red due to the low iron content. There's another form of anemia called thalassemia, and this is common in individuals of Mediterranean descent, and it's due to an increase in red blood cell destruction. Thalassemia is a genetic disorder where individuals suffer from a defect in the cytoskeletal membrane, and these cells then are more likely to break down. The next form of anemia we'll talk about is sickle cell anemia. It's a genetically determined defect of hemoglobin synthesis that results in hemoglobin instability and insolubility. What happens here is that cells become sickled, which leads to vascular occlusion that can be painful. It's a more severe form of anemia, and the red blood cells are different shapes and sizes. This causes painful episodes due to vascular occlusion. Now, what this looks like then is that when red blood cells are oxygenated, they have sort of the more normal biconcave disc shape. However, when red blood cells have low O2 levels, you get odd conformations or changes in the shape of hemoglobin, which also changes the entire shape of the red blood cell, and it gives it more of a sickled shape. So with sickle cell anemia here, as blood starts to deoxygenate, these erythrocytes will sickle. They're unable to pass through smaller arteries, and cell membranes become damaged, and therefore the red blood cells have a shorter lifespan. Now, this leads to anemia because these red blood cells have a sickle shape. They're more likely to become damaged, and therefore each individual red blood cell has a shorter lifespan. Now, this can lead to things like occlusion of small arteries, which can cause tissue damage, pain, and loss of function. It can also lead to increased hemolysis or breakdown of red blood cells by the spleen. This can cause damage to the spleen and also lead to what we call hyperalbuminemia. And so this is actually going to be where you have, hyperbilirubinemia, which you have too much bilirubin in the bloodstream. And bilirubin is the breakdown product of hemoglobin. And if you get too much hemoglobin in the blood from increased red blood cell breakdown, this can lead to jaundice. So one of the signs of sickle cell anemia can be jaundice due to an accumulation of bilirubin from increased red blood cell destruction. The last one here is hereditary spherocytosis. This one's a defect in red blood cell membrane skeletons, and this is going to be where the cell red blood cells are more fragile. This is also heritable, and it's actually autosomal dominant. So you only need to inherit one defective copy of the gene that encodes for this red cell membrane skeleton in order to have hereditary spherocytosis.